Okay, great. We're live on YouTube. Yeah, people sign up last minute. I think they're used to there's so many fancy versions of Zoom where you get like an auto registration and it sends you the link magically right away. But that's for the Zoom you pay for, right? Not the kind of Zoom that we have. So we don't have like a staff monitoring all this. And so I feel bad because I know there are some people out there like, oh yeah, we need to sign up right now. And I'm like, sorry, I don't have time to send you the link. So hopefully most people, if they didn't get it in time, will be able to catch up on YouTube afterwards. And we do our best. There's like 3,000 people on the mailing list. So I'm like, all right, close enough, y'all.
The regular people know where we are. They always manage to find us.
The people who show up every week. Yes. All right. So I think maybe I'll just wait like 30 more seconds and let a couple.
Thank you so much, Sara, for accepting our invitation. This is like the end of our season. So we're going out with a blast.
It's an honor to be the closing of the season.
Yeah, I like that. We're like, yeah, finishing strong. We had a lot of new people sign up for the mailing list after we advertised your talk. So that's always that's nice to see lots of excitement.
I'm sure we pulled some people who are not like hardcore, so like, migration people, more mechanics people.
That's right. Well, lots of overlap, right? Cell migration's at the center of it all. All right, great. I'll go ahead and get started, and more people will continue to pop in. So hello, everybody, and welcome to our final cell migration seminar of fall 2022. We do plan on being back in the spring. If you're interested in presenting, reach out to us. We've got the very old, poorly maintained website that you can submit a abstract on, or you can email either myself or Ankita or both of us, and send us an abstract, tell us what you want to talk about, and we'll start thinking about that in January probably. So welcome everyone. We are so delighted to have our final speaker of the season, Professor Sara Wickström. So Sara received her MD in 2001 and then her PhD in 2004 from the University of Helsinki in Finland. She did postdoctoral training at the Max Planck Institute for Biochemistry, and after that she became a group leader there in the biology and aging in 2010. And then in 2018, her lab moved back to the University of Helsinki, and she was a professor of cell and developmental biology there. But now just this year in 2022, Professor Wickström was appointed as director of the Max Planck Institute for Molecular Biomedicine in Münster. And so research in the Wickström lab aims to understand how mammalian epithelial tissues are generated and maintained. And in particular, they focus on how mechanical forces and cellular interactions integrate single cell behaviors to pattern structurally extremely robust yet dynamic tissues. So it's such a fascinating question that we're all really interested to hear about what you guys are up to. So with that, I'll hand the floor over to you, Sara. Take it away. Thanks so much.
Great. Thanks so much, Jennifer and Ankita, for, first of all, hosting this whole series. I'm a big fan, and of course for the invitation. So it's a pleasure to have the opportunity to present some of our recent work. So as Jennifer said, my lab is interested in how
Sorry, are we losing her on your end too?
I don't hear.
Yeah, Sara, I couldn't hear. We heard you right in the beginning and then you cut out. Can you still hear us?
Can you hear me?
Yes.
Yes.
Yeah, we lost you like for 2 seconds.
Weird. Okay, I better actually, let's see. I get the message that my internet is unstable. I'll just plug in. Can you hear me?
Yeah.
Yes, you're perfect.
We'll let you know if there's any issue.
You're gone again.
Sara, we lost you again.
Oh, I'm sorry.
Of course, it's for our last
Take it.
one that we'd have technical problems. Is there any message showing up on your screen or?
Yeah, just that the internet is on. I think the wireless is a little bit poor, but
okay.
let's
And now
hope
we
that-
can hear you again. So
Okay,
somehow the
well,
Wi-Fi knows right when you talk about science. I don't know.
yeah, exactly. Yeah, I'm really sorry about that. Usually
No
I
worries.
don't have these kind of issues.
Keep
So
going.
yes, so in terms of information transmission across scales, particularly interested in that and how the cells really, from the molecular perspective, establish their identities, how they maintain them, and how these cells with different identities communicate with others to coordinate the fate and their position within groups of cells, and how these groups of cells then go on to organize into these highly complex tissues. And of course, we know that this tissue architecture feeds back all the way again to this regulation of the cell states. But how do these scales cross? It's not so well understood. And one of our favorite organs to ask these questions is the mammalian skin epidermis. And in many ways, it's a fantastic organ system to study dynamics and cell fate coordination. And one particularly interesting time point is the early embryonic development, on which I will focus today. And that is that in the mouse, this is embryonic day 12 to 13, where basically from the single layer of surface ectoderm, two events happen simultaneously. And this is the establishment of the stratified epithelium termed the epidermis, but at the same time, the same cells will go on to make the hair follicle, which is a completely different and kind of independent organ within tissue. So the question is, how is this symmetry broken from this homogeneous layer of ectodermal cells, where then single cells exit the stem cell state and then start moving upwards to form these multiple layers, which each layer represents a further differentiated state. And at the same time, the same cells enter a completely different fate of the hair follicle stem cells and start moving downwards and invaginating to then make the mature hair follicle. And in particular, in the epidermis, the dynamics actually continues throughout our lives, where you have this dynamic turnover of the tissue. So the epidermis is actually one of the fastest turning over tissues after the intestine in our bodies, and actually millions of cells are shed every day. So this kind of dynamic coordination of cell fate and position is a kind of a requirement of the homeostasis of the tissue as well. But today I'll focus on really this early embryonic developmental stages to ask, how is this symmetry broken? What defines these cell fates, and how are these cell fates coordinated with the position as the dynamic continue? And this is really work of started by Katie Miroshnikova in my lab, and then taken over by Clementine Villeneuve and Ali Hashmi. And it's been a fantastic collaboration with Lisa Manning, who is a physicist and a modeler, and her amazing PhD student, Liz Lawson-Keister. So Kate, being an engineer, wanted to build a very simple system to study this kind of dynamics of differentiation, cell fate commitment, and stratification. And luckily enough, actually, we can easily culture the epidermal stem cells in vitro and induce differentiation just by adding calcium to the cultures. And what will happen in this very simple 2D plastic culture is that single cells will exit the stem cell state, terminally differentiate, and form a second layer. So this is a kind of a very reductionistic model of stratification. And she decided just to kind of watch what happens as this stratification proceeds. And it was quite surprising for us to see that there was a very dramatic transition in terms of the tissue scale mechanics. So in the undifferentiated state, the monolayer behaved like a fluid, so there was this kind of large-scale coherent flows. But then around 24 hours after application of calcium, this kind of flow became completely halted. So there was this very abrupt jamming or glassy transition in this system. And what was interesting to see was that although cell divisions themselves were not halted, so this was not really contact inhibition of growth, but rather this kind of mechanical jamming transition, at that point we started seeing delamination. So at that point, the cells started moving upwards as single cells. And what was interesting to see was that at that point, we often saw that these delaminations also happened in close proximity of cell division. So soon you will see few cells dividing here, and then their kind of close proximal neighbor was moving upwards. So of course, this was a very simple system. So we first wanted to ask, what is kind of the mechanical state of the actual in vivo mouse epidermis? So here you can see now this delaminating cell. So what is the in vivo state of the epidermis in the mouse? And the nice thing of working on the skin is that it's on the surface, so we can actually take embryos out ex vivo, and the skin will continue developing and growing for around 12 hours. So we can place the embryos under the microscope and really image live as the stratification proceeds. And what we noticed was that at that point, at E14.5, E15.5, that the tissue was in this jam state as we observed in vitro. And what was further interesting to note was that, again, we saw this kind of close proximity of cell divisions and delaminations. And when we looked at these regions where we saw this close proximity, we often also saw this kind of anisotropy of the tissue. So basically, the cells showed more elongated shapes, and the axis of elongation was in one direction, suggesting that there's some sort of tension anisotropies at these areas where we had this increased delamination division proximity. So of course, these were two correlations. So we had this correlation of the jamming transition and the proliferation and the delamination. So we hypothesized that maybe this kind of cellular crowding could actually be triggering, in fact, the differentiation. And to test this experimentally, Kate Bain went back into the in vitro system and took the epidermal stem cells and cultured the non-silicon elastomers that were statically stretched to 120% of their original size. And then she abruptly released the tension to kind of experimentally introduce crowding. And indeed, if we do qPCR of the differentiation genes of the system, we can see that this kind of experimental crowding is really sufficient to actually trigger expression of the differentiation genes. And in fact, if we do the same experiment with subconfluent cells, we don't see this. So it's not an effect of substrate buckling or something, but really a function of cell density, further supported by the notion that even in this kind of steady state cultures where we don't do the experimental color crowding, the expression of the differentiation genes really scales with the density of the monolayer culture. So increased cell density seemed to be a trigger to push cells towards differentiation. So then we wanted to ask more precisely what is actually kind of the geometrical trigger and what is the relationship between these force anisotropies that we saw in vivo and the differentiation. So to test that, Kate turned into kind of a different model of crowding where she engineered these kind of micro patterns where we can force the cells really to adhere to any geometries that we generate with the UV lithography. And she basically generated two types of geometries, circular and squared geometries. And these islands have exactly the same surface area and they host approximately the same amount of cells. But we hypothesized that this crowding that is induced by the pattern is a little bit different because these squared geometries obviously have the corners, and we hypothesized that the corners will actually push the cells to elongate, which indeed is happening, whereas here on the circles, they have more isotropic shapes. And in addition, they actually, by traction force microscopy, we can approximate the strain anisotropy of these cell clusters, and indeed, as also predicted, the cells on squares show higher degree of strain anisotropy. And indeed, just this subtle change in cell geometry actually increases the probability of cells to differentiate. And additional experiments that I don't have time to show kind of made us then conclude that the most powerful trigger, like geometrical trigger for differentiation, is restricting the cell matrix adhesion area, but at the same time forcing the cells to elongate. So really kind of producing these restricted but elongated cell shapes. And we have meanwhile been very interested in how these signals are propagated into the nucleus and how these differentiation pathways are triggered. And this is kind of a very important and central research focus in my lab. But since this is a migration seminar, I will not go into too much detail. I'll just summarize a lot of work in this slide. So we have started to understand how actually cells sense deformation and how this affects cell shape. So basically, for example, by doing experiments with confinement and the opposite, so basically stretching, so increasing tension, we see that this is in fact kind of globally affecting RNA polymerase activity. And this allows then gene silencing to access the lineage specification genes. So for example, the polycomb repressive complex 2, which is a repressive histone methyltransferase complex, can come in and then silence its specific target genes, because there's this kind of competition of promoters. The transcriptional machinery and the silencing machinery, they kind of compete. And if we downregulate transcription, this gives the polycomb an opportunity to come in, silence the differentiation genes. And what this does is then this kind of reduces the probability of differentiation in this kind of relatively unspecific way. So basically our current working hypothesis is that if the cells are under high tension, for example, if they're stretched or cultured on stiff substrates, they have low level of gene silencing, and thereby their differentiation probability is lower than cells that are being laterally confined. They have low tension, lower levels of gene silencing. So it's kind of a probability game by which cell geometry can instruct cells to respond to a biochemical differentiation signal. But when we were doing these micropattern experiments, we observed something unexpected. And this was that we also saw this fluid to solid-like transition. So you can see here this micropattern, the cells are flowing, but then they transferred into the jam state, but they did it in a very stereotypic geometric arrangement where always one single cell ended up sorting into the middle. And what was further intriguing about this behavior was that the center cell was invariantly always the differentiated cell, which ended up delaminating from the middle. And of course, this kind of behavior is classic developmental biology, so cell sorting. So people have worked out a lot on the cellular mechanisms, and we know that cell sorting can be driven by differential cell adhesion, cortical tension, both now also even membrane tension fluctuation. So we knew to kind of expect that this differentiated cell should have differential cell mechanics. And we also noticed that the kind of the cell fate commitment happens first and then the sorting. So we hypothesized that basically these mechanical changes follow the cell fate change. And indeed, if we measure the mechanical properties of differentiated cells, for example, using AFM, we saw that there was this interesting biphasic evolution of cortical stiffness. So initially at the early stages of the differentiation, the cells became softer and then they regained their stiffness. And when they delaminate, they are actually stiffer than the basal cells. But at the time point when they are differentiating, they become softer. And then at a little bit later time scales, they actually also increase their cell-cell adhesion force. And this increase in cell-cell adhesion forces, we think, is driven by multiple mechanisms, for example, also by the desmosomal cadherins, but we also saw that the kind of the surface expression of E-cadherin is also increased at this time scale. And indeed, if we delete E-cadherin, we no longer see such a strong increase in the cell-cell adhesion. Whereas surprisingly, at these early stages, if we measure cell matrix adhesion, it's not so much change. So we hypothesized that the changes in the cell-cell adhesion actually precede the changes in cell matrix adhesion. So to test whether these mechanical changes that now coincide with the cell fate commitment could participate in coupling the cell state change to the cell position change, we turned into a little bit more elaborate stratification assay, so-called organotypic cultures, where we can produce a little bit more in vivo-like epidermis. So we embed fibroblast into a 3D matrix of collagen, and then we let keratinocytes adhere on top, and they will really form nicely a single layer of stem cells and then multiple layers of differentiated cells. And then to test whether this mechanical change is required to couple the cell fate change to the cell position change, we depleted E-cadherin because we knew that was the driver of these mechanical changes, and we mixed E-cadherin knockdown cells with wild-type cells 50-50, and we also labeled the E-cadherin cells in green so that we could trace them in the tissue. And what was surprising was that even just 50% of the knockdown cells was sufficient to perturb the overall architecture, and we didn't have a massive delamination defect. But what was clear was that now the coupling of the state and position was lost. So we started seeing differentiated cells that remained in the stem cell layer, whereas some stem cells seem to be delaminating, really suggesting that this kind of robust coupling of cell fate change to the cell positional change requires the change in cell mechanics. And of course, this is an intriguing question, and now we are trying to address this in vivo and also in silico, because the question is now, is this kind of change is sufficient actually to drive delamination? So in collaboration with Lisa Manning, we have been exploring 3D vertex models of the epidermis, and this is very much work in progress, but Lisa has generated a 3D vertex model where we basically model the basement membrane as basically an extremely stiff layer of cells, and then we have a network of springs between the basement membrane structure and the basal cells, and then we have suprabasal cells. And in this system, we can now start modulating basically the interfacial tensions between cell layers, but also within single cells, and start asking whether these mechanical changes are really sufficient to drive cell movements. And indeed, in this very preliminary analysis here in this red cell, we have now started to change the interfacial tensions, similar to what we see basically in vivo. So we changed the lateral interfacial tensions as well as the apical, and then we can see that these kind of decreases in interfacial tension are actually sufficient in this model to drive delamination. So we are very excited about this experiment, because it suggests indeed that this kind of local mechanical differences at interfaces are critical to reposition cells. And of course, at the same time now we're doing similar experiments in mice where we try to generate mosaics where we perturb really the mechanics of a single cell, for example, by deleting E-cadherin or myosin, and seeing whether this is sufficient now to have these specific cells delaminate. But the current model for the early epidermal stratification is that this increased proliferation early on drives the jamming transition, which then triggers lateral confinement, and thereby the cells that are most confined are the ones that then have a lower threshold for differentiation in response to a biochemical cue, and this drives cell fate commitment. And then this commitment is associated with the changes in interfacial tension driven by changes in cell-cell adhesion and cortical mechanics, which then ensures that this differentiated cell is also delaminating and positioning correctly into the suprabasal layer. But what about the hair follicle? So this is now an unpublished study, so I'm very excited to hear your feedback on this as well. So this, since the other study is already a few years old, so meanwhile we have really been focusing on establishing the tools that we had in the in vitro work to really have them working in our embryos, and particularly because unlike the epidermis, which we can fantastically culture in vitro, the hair follicle is a more complex system, and we don't have so good in vitro models for that. So we are also relying more really on the in vivo systems for this work. So what we started off was with a very similar characterization that we did for the epidermis to try to understand what is the relationship between cell fate and cell mechanics, as using cell shape and kind of tissue scale parameters as proxies. So again, we looked at the early embryonic development, and FGF20 is the earliest or one of the earliest known markers of the hair follicle compartment. And then we used the reporter mice that express beta-galactosidase under the FGF20 mode, so we can see the single cells that start expressing FGF20. And at D13.5, you clearly see these kind of pre-patterns of these early hair follicle cells forming in the epidermis. So this is a top view of the actual embryo. And interestingly, how carefully we look, we actually do not find any cell shape differences between these cells and the neighboring interfollicular epidermis. So this strongly suggests to us that the early cell fate change precedes any mechanical change in the tissue. And then remarkably, just one embryonic day later, you can see that now the cell clusters are larger and stronger. We see this kind of quite substantial cell shape change. So in XY, you have this decrease in XY cell area. And then you can see also that the cells change their, or the tissue scale nematics change. So you can see this kind of elongation around the pattern and also changes in the cell shape. So this structure here is called the placode. So that's kind of the early epidermal thickening. So during this one day, you have this quite substantial change in the cell shapes. But it seemed, and this is also work that is done by multiple labs in the field, that the initial commitment of the cells is actually driven by signaling rather than mechanics. And it seems to operate a little bit through this kind of Turing-like patterning and signaling. And we know that actually the mesenchyme, so the fibroblasts in the dermis, are critical for the hair follicle specification, and they actually seem to secrete a Wnt activator that initially produces a broad distribution of Wnt, which then through this kind of Turing-like mechanism where you have short-range activator and long-range inhibitor, then restricts and forms this pre-pattern of the cells. But we then were very interested to understand what really then drives this very rapid cell shape change directly after this early commitment. And we first kind of tried to describe very carefully what's happening in vivo by doing 3D segmentation of the cell shapes, and the initial transformation between this E13 cell fate commitment and the thickening seemed to be like a volume preserving elongation. And interestingly, there were no signs of apical constriction, although you have this kind of thickening. It was really an isotropic elongation where both the apical and the basal surfaces were becoming smaller proportionally. And then to understand what kind of mechanical changes on the tissue scale are associated with this transformation, Clementine went back into the ex vivo embryonic imaging and started analyzing the tissue scales flows. And here you can see really the E14, you can clearly identify the placode based on this apical, this XY cell shapes here. And I don't know if you can see this PIV analysis very well, but basically what we see at this stage are this kind of centripetal oscillatory flows of the tissue. However, if we measure the mean divergence of the PIV vectors, so kind of the net direction of the vectors, we don't see any big changes, and this is also quantified here. So it really seems to be kind of this kind of non-productive oscillations, if you will. However, at this point, the tissue, this placode is under tension, also confirming that these changes are most likely driven by actomyosin tension. So you can see that basically this is the photoablation and the radial displacement of the epidermis and the placode. So you really have this recoil in both directions, showing that the tissue is under tension at this point. And interestingly, one embryonic day later at E15.5, what we now see that these oscillations basically continue and even become a little bit more intense. But now we start seeing actually net negative divergence. So basically this suggests that there's some sort of sink of these flows downwards. And of course, this is a Z scale invagination. So it makes sense that now this tissue actually starts moving in depth into the dermis. And indeed, if we basically look at this from the side, you can now see that this tissue really starts flowing downwards. And we see that this deformation is basically partially plastic, so it's no longer elastic, so basically returning after these oscillations back into the original shape, but we really have this kind of net downward flows as also predicted by the divergence analysis. So there seems to be this initial elongation process that is potentially driven by this isotropic oscillations, and then in the second step, so one day later, now you start having these tissue flows downwards. So the question is, what is driving these flows? And obviously, actomyosin is the prime candidate also because we show from the laser ablation that the tissue is under tension in this area. But looking at the myosin distributions, we were actually quite surprised to see that in fact, in the placode, so in the kind of the site of the invagination, we actually have very low myosin activity. We see high myosin activity in the apical surface and in the suprabasal layers, but the placode itself has low contractility. However, interestingly, if we go deeper into the tissue, and this is a side view, but this is now a top view, we see that actually there's a ring of fibroblasts that gradually assembles around the placode. And these fibroblasts are also contractile and much more contractile than the placode itself. So you have now two potential sources of forces that are acting on the placode cell. So we have this high apical contractility, and then we have this ring of fibroblasts. So we again went back to Lisa and asked whether she could somehow do an in silico experiment for us to really probe what would be possible mechanisms, how this kind of deformations could form, and whether we could probe in silico kind of the possibility of cell autonomous forces driven maybe by changes in the lateral interfacial tension because this area has low myosin, or basically high apical tension because we have high myosin here, or by extrinsic forces driven, for example, by the contractile fibroblasts. So basically we focused on these three parameters, which was the ratio of the stiffness above the placode and below the placode, then these interfaces between the placode cells, as well as these extrinsic forces. And we used the same 3D vertex model that I already introduced to you. And now we can start basically producing these kind of phase diagrams where we really simulate a large range of parameters. And then what is cool about this in silico experiments that we can then measure actually the deformation of the tissue, and then we can directly benchmark these simulations to the actual measurements. And here I'm just showing one phase diagram where we have looked at the curvature, and you can see how the simulations actually here, for example, this is now the apical to basal surface tension ratio, and this is the external force. We can see that basically high external force together with basically a high apical to basal surface tension ratio, which is basically recapitulating the myosin distribution, it actually forms something that very closely resembles the placode. And of course, we don't have to rely on the visual inspection, but we can really look at the data. So here we have the two stages. So the thickening and then this kind of downward flowing. And we can see that indeed this extrinsic force model much more kind of covers the actual parameter space of the actual measurements, suggesting already that probably the extrinsic forces could be relevant here. And indeed, if we then also do in silico PIVs to understand the tissue flows potentially generated by the different forces, we can see that only the extrinsic forces are actually able to generate this kind of negative divergence that we see at E15. So this basically made us conclude that possibly this model 1, so the changes in the lateral interfacial tension, could be sufficient to recapitulate some of the early stage changes, but this production of the full mature budded placode that is characterized by the net downward flows most likely could rely on these extrinsic forces. And to test these predictions from the model, we then decided to delete myosin 9 because we know from our experiments that myosin 2A, so basically the heavy chain encoded by the myosin 9 gene, is really the major myosin in the epidermal cells driving actomyosin contractility. And if we do this in the epidermis, we indeed basically see that there is an effect. So there is an effect in that the placode is not producing this nice deep curvature and also invaginating down. And also if we look a day later, we see that basically this is the epidermal compartment and this is the dermal compartment. So this fibroblast ball that is directly underneath the placode. And it seems that this epidermal myosin is really required to kind of produce force against this dermal compartment to really push the placode downwards. But it seems not to be really essential, first of all, and secondly, not essential for the full cell shape transformation. So actually the individual single cells are actually quite well elongated even in this knockout. And surprisingly, if we now delete myosin in the fibroblast, so we use twist 2 Cree because these fibroblasts are high in twist 2, actually the phenotype of the placode is much more severe. So we see, first of all, that the myosin is required to the full assembly of this fibroblast ring. And now we see what we didn't see in the epidermal knockout is that actually we start seeing less placodes overall, so really suggesting that the dermal myosin is essential for the placode formation. And of course, if we now interfere with myosin in both compartments by taking explants and treating them with blebistatin, we basically completely abolish placode formation. So contractility is required, and it's required in both compartments, but it seems that really these extrinsic contractile forces are even more important for producing the full placode. And what was surprisingly and completely unexpected, that if you look at this dermal knockout, so now we have deleted the myosin only in the fibroblast and not affected the epidermis itself in genetically. And now if you look at SOX9, which is a later master transcriptional regulator of the full mature hair follicle stem cell fate, we see that this SOX9 doesn't become compartmentalized in the placode. So it really kind of is scattered around, and we have fewer SOX9 positive cells. So this made us think that maybe this kind of these extrinsic compressive forces are also required to compartmentalize the SOX9 expression into the placode. And to test this experimentally, we took the epidermal cells, and if we put them in 3D, we can actually maintain them for a short period of time as hair follicle cells. And if we now basically exert hydrostatic pressure on these hydrogels by basically exposing them to static compression, we can see that this enhances SOX9 expression, really showing that confinement in this kind of hydrogel system, also very similarly to the interfollicular epidermis, is basically driving differentiation. But of course, the question is, what then facilitates these downward flows? What allows the tissue to kind of fluidify and start flowing downwards at this later stage? And when we started looking at this in imaging data more carefully, we noticed that at this stage, actually, we had very many cell divisions in the placode itself. And this made us read the literature of other epithelial invaginations a little bit more carefully. And we came across, for example, this paper from 2013, where they showed that in the Drosophila trachea, local cell divisions actually promote the invagination of the trachea. And we started thinking that maybe these cell divisions, localized cell divisions here in the placode also play a role in this fluidification and the downward flows. And indeed, when we look at proliferation, it shows a very interesting pattern, really, again, showing this day-to-day switching of very, dramatic cell states. So at E14.5, when we have the thickening but no downward flows, we actually have no proliferation in the placode. And in fact, also nuclear YAP is excluded. And just one day later, now we have massive local cell divisions and also re-entry of nuclear YAP here locally. So this suggests, and based on also work from others, for example, Aurélien Roux has beautiful work recently in confined in vitro epithelia, showing that basically if you confine epithelia in these alginate capsules that he uses, this actually excludes nuclear YAP and prevents proliferation. But when this pressure then in the end allows the epithelium to buckle, this allows YAP re-entry and proliferation. So we started asking that whether in this case also the re-entry of YAP through maybe release of pressure is allowing the re-entry of cell divisions. And indeed, if we confine the cells, we can recapitulate the exit of YAP from the nucleus. And if we treat our explants with verteporfin to block YAP, indeed these cells re-enter the cell cycle, which basically recapitulates a lot of observations for others. So we think that this local confinement at E14.5 excludes nuclear YAP and prevents cycling. And then there's some sort of release of pressure, which then allows the cells to enter the cell cycle, possibly mediated by YAP. But of course, the question is first whether these cell divisions are important for the placode to proceed to make the full budded structure. And for this, we retorted to explants. So we take the skins from the embryos and we can culture them actually for several days, and the hair follicle morphogenesis will proceed perfectly. And then we did a short pulse of mitomycin for 3 hours and let the development proceed in these cultures. And indeed, blocking the cell divisions really block this further development of the fully budded placodes, suggesting that localized cell divisions are most likely important for the full maturations. But of course, the question is what would allow the release of compression? And to understand this, we started looking at the basement membrane, because of course there's this specialized, very stiff matrix structure that separates the epidermis and the placode from the dermis. And to our surprise, actually, we didn't see much in terms of components being differentially present, or even that the basement membrane is clearly there, and it actually looks pretty similar than around the placode. However, if we measure the stiffness of the basement membrane from cryosections using AFM, we see that especially at E15.5, the basement membrane below the placode is much softer than that of the epidermis. So we started thinking that maybe it is locally actively remodeled at this stage. And to ask whether this is the case, we reanalyzed single cell sequencing data set from the embryonic stage E15, published by Gupta et al. And indeed, what was curious to see that this is the placode population here, and this is the dermal condensate, so the fibroblast population directly beneath, and they are very high in MMP14, which is this membrane-bound MMP. So it really suggesting that there's probably localized proteolysis just basically at the interface of the fibroblast and the placode. And the fibroblasts are also very high in MMP2. So this suggested that there's localized proteolysis at E15.5 at this stage. And to ask whether this MMP activity is required, we again went to our explants, treated them with MMP inhibitors, and we can quite substantially actually block the placode invagination, suggesting that really you need this matrix remodeling to kind of fully release the pressure and allow this downward growth. So our current model is that, unlike in the epidermis, it seems that these early cell fate changes precede any kind of morphological changes in terms of the hair follicle, but then very rapidly these cells change their mechanical properties. They produce this kind of isotropically elongated shapes, and this basically is associated with the kind of the initiation of assembly of this ring of contractile fibroblast around the placode, which we think is building up local compression and prevents YAP from entering the nucleus. And this pressure is then subsequently released by localized basement membrane remodeling, which then basically allows YAP nuclear re-entry, localized cell divisions, which then produces this kind of fluidified and downward flow of this structure. Now, to put things into context, I've been reading the literature, and kind of recently there's been some amazing studies really kind of showing similar things in various systems and in various basically model organisms, really raising the common theme that cell density is controlling the tissue scale mechanical properties in many scenarios, which then drives morphogenesis. So there's work from Roberto Mayer, Otkar Campos showing really that this kind of tissue solid jamming transitions are driving morphogenesis. And there's also this very interesting recent study from Edward Hanezo showing this kind of rigidity percolations that also drive embryonic development. So this makes us think, coming back really to the bridging of scales, that one way to really convey information across scales are these mechanical tissue scale changes, where cell density changes and packing changes drive these kind of fluid to solid transitions, which then communicate basically to groups of cells that then change their shape and adhesion, interfacial tension, to then again change their position in response to these cues, but also allowing single cells to basically cross compartments in a mechanical jam tissue. And then finally, these kind of local cell shape and adhesion changes drive intracellular responses, where changes in the contractile cytoskeleton and nuclear shape and deformation then globally tune transcription and chromatin architecture, which then changes the way the cell interprets biochemical cues and thereby drives differences in cell fate and state changes. And with this, I'll close and just thank all the fantastic people with whom I had the privilege to work over the years. So much of the work I presented here was done by alumni who have now already left the lab, but also by Clémentine Villeneuve, was really pioneering the placode project. And we've had amazing collaborators without whom this would not have been possible, particularly Karin Nielsen, and of course, Lisa Manning, with whom we collaborate on the simulations. And with this, I'll close, and I'll be happy to take any questions.
All right, thank you so much, Sarah. That was fantastic. And we've got a lot of times for questions, which is great. So let me just scroll back up. Our first question is from Anna Pasapera. And so this was from a little bit earlier in your talk. And she was asking if you measured if there were changes in the protein composition of the extracellular matrix, or if there was growth factor release, because with cell crowding, that could maybe induce more differentiation. I think that's
So I don't know whether is it maybe related to the first part or the second part. So for the first part, we have not done it, and I think that's a very cool question in that respect. So whether there would be still localized changes in the basement membrane, for example, at the sites where the cells are delaminating. So that's something very interesting that we are actually addressing right now. With the Planck code, we've looked quite carefully, and we haven't found any components that would be obviously different. So we really think that it's just kind of turnover, localized turnover, where the cells are kind of putting down the basement membrane, but also remodeling it continuously. And that's why you need these proteolytic enzymes to basically release pressure. So the basement membrane is there, it has the same composition, but it's just being turned over faster.
Awesome. Ajay, will come to you next. I'll take a question from Mohammed Haroon. He's asking, did you guys perform some sort of membrane-specific proteomics with different cortical tension-treated cells? He's thinking in lines of seeing the changes in cell-cell adhesion in terms of this altered membrane proteomics.
Right. Yeah, we have not done that. So that's, of course, very interesting in terms of whether, because there's this intimate crosstalk between membrane tension, membrane composition, and cortical tension. So I think this is fascinating, but we have not really looked yet whether there would be some sort of changes that we would associate with the changes in the cortical tension.
Awesome. Ajay, if you want to go next, please go ahead.
Yeah, that was a great talk, and it's actually beautiful because there are many parallel themes in the primordium, which is sort of derived from a placoidal structure and continues to pattern itself. But so my question was this important role of the fibroblast ring in the dermal layer. How much do you think that is in response to mechanical changes in the aggregation of cells, right? Which they surround, versus signaling that has triggered this process, or what's the interplay between those two things?
Right. That's a great question, and I don't have an answer. So my guess would be that it could be both. So we don't know what actually. So obviously there is probably a signal coming from the epidermis that recruits these cells to form this ring, but what is also interesting is that there's also actually a ring of blood vessels there. So it could also be signals from the circulation even that is recruiting these cells. But of course there could be then once the ring is there could be mechanical crosstalk, because if we basically analyze the fibroblast, we see that they are also oscillating. So I mean, we need to still do some work, and we haven't, for example, done the laser ablation of the fibroblast, and to see whether we would then see, for example, relaxation. So we need to do some additional characterization, but I think that's very interesting.
Thanks. Great talk.
Thanks.
Thanks, Ajay. I'm going to skip a couple questions because it looks like there's a follow-up from Mohammed Haroon's question. So just to go to that. And he's saying, when you mention the role of actomyosin contractility, do you mean the actomyosin of the cortex compartment of the cells, or are you thinking do you mean the cytosolic compartment contractility changes?
I didn't quite, sorry, I didn't.
Yeah, Mohammed, if you want to clarify, but I think so. It's, we can have cortical actomyosin, or we can have, actomyosin structures elsewhere in the cells. Is that what you meant, Mohammed?
Yeah, exactly. So what I mean actually is the cortex of the cell and the cytosolic part of the cell, they both have like different actomyosin contractility. So when you mention the changes in actomyosin contractility during differentiation and this whole process, can you do you have any insight into the data differentiating between the two? Is it cortical? Is it cortex, or is it overall cell contractility?
Right. Yeah. So you are referring to the first part where we saw this kind of biphasic. So we know that it's driven by myosin activity, because if we delete myosin, we first of all we lose basically the cortical stiffness, and we also lose the cell sorting. So that is also required, basically, these differences in myosin activity. But whether now this myosin is acting on kind of the cortical actin or more, let's say, the stress fibers, it's difficult to address. These cells in general don't have so many stress fibers because they're epithelial, so the actin is mostly cortical, but I don't think we can really conclude that what is kind of the major pool. So myosin is required, but on what pool it's acting, it's kind of difficult for us to dissect.
Thank you for addressing.
Thank you so much, Sara. Next question is from Luke Bolter, says really beautiful work. And saying that the formation of plaque is very reminiscent of the early phases of budding morphogenesis and the deformation you see there. How universal do you think isotropic oscillations are in breaking symmetry in epithelial sheets or tubes?
Yeah, that's a great question. And indeed, we are not the first ones to report. So, for example, Celeste Nelson has beautiful work showing this kind of process. So I think it is probably a commonly used mechanism to actually elongate tubes. But what is interesting is that at E14, we see these oscillations, but there's no net elongation. So in our case, you seem to still require further this local fluidification to have really productive elongation. And that might be really dependent on kind of the environment, because we have this stiff, dense connective tissue of the dermis below, but maybe, in tissues where you have an epithelium that is not directly overlaying connective tissue, maybe then you don't need this kind of mechanical transition. So I think it will be very interesting to kind of further compare various systems.
Okay, great. We have a question from Yanin Guerrera asking about how are you measuring the stiffness of the extracellular matrix, and are you measuring it using an elastic model?
Right. Yeah. So we, did it the best way we can in this system. So, which is obviously, has its limitations. So we use a cryosection, and then we use a spherical AFM cantilever, and then we fit the force distance curves to a Young's modulus. So, obviously the method has its limitations, but we still believe when we are comparing different things, for example, the placode and the surrounding IFE from the same structure, and we see differences, we think that we are somehow kind of detecting true changes in the elasticity. But we are currently kind of exploring additional ways by which we can probe the mechanics also in kind of more intact tissue, and not having to revert to this kind of methods.
Awesome. Next question is from Radhika. Since I see Radhika here, would you like to ask your question?
Sure. So, Sara, beautiful work. So nice to see you direct such gorgeous work in the pandemic. So my first question is, have you considered using earlier Cree lines for the dermal side instead of twist 2 Cree? Because twist 2 is still a bit later than some of the earlier Cree lines. And simultaneously, what about using something that's not beta-gal driven, such as the FGF20, which has its own limitations? Maybe some earlier lines that are live reporter systems, like Axon 2 GFP or something like that.
Right. Yeah, these are great suggestions. So we are actually working on that right now, because obviously we also want to, the LAC-Z we are doing in fixed hole mounts. So we want to obviously look at earlier stages. So we are actively constructing CRISPR reporters for earlier stages. We see the twist 2 coming in expressed at this kind of E14, E15, but I agree. I think it would be cool to do a deletion even earlier. So thanks for the feedback. I think these are both really great suggestions.
I think it might, I don't know if it would cause more trouble, because it might affect the morphogenesis of the dermal fibroblasts, because they're also under mechanical forces.
Right.
So I don't know if messing with them too earlier would cause trouble.
Yeah.
But gorgeous work.
Thanks. And good to see you. I think we
Great
haven't
to see
seen in ages.
Great. Thanks so much. We have another question from Anna, who says, wonderful talk. And did you take a look at the initiation of the placode formation, specifically for if besides the actomyosin contractility, were there also changes in the centrosomal and mitotic spindle positions, which then could also contribute to reorganization of the tissue?
Right. Yeah, that's a great point. Indeed, we are ignoring so far both the intermediate filaments as well as the microtubules. So we definitely should look at both of those also. But the fact that we really tried hard to see any shape changes, we kind of conclude, but I think we should look at the other cytoskeletal components indeed more carefully.
Sara, I had like a naive question. I understand that the initial placode formation was because of the signaling, as you suggested. But later in the stages, when you have these changes in the, in the tension, is it purely, it can't be purely mechanical, but what's really, sort of upstream regulation is like there?
Right. So basically our hypothesis is kind of difficult to show, but it's kind of the theme that we gather both from the epidermis and the placode is that you have signals that specify cells, but these are regulated by the tissue scale factors such as cell density. So, but they don't actively switch cells from one state to another. They kind of more kind of instruct the probability. But then these state changes are associated with changes in cell mechanics, and how this happens, we can only speculate, but obviously it could be transcriptional regulation of actin, myosin, the components of this.
But also adhesion, right? Because that's such precise changes in cell adhesion.
Exactly. And I didn't go into this, but actually we see very specific patterns of cadherins. So what is really interesting is that in the basal layer, you have both E and P cadherin, but actually in the placode, they have very high P cadherin and lower E cadherin. So definitely these kind of changes in the cell adhesion are associated with this kind of cell elongation that we see. So we think that kind of state changes drive changes in adhesion, actomyosin, cytoskeleton, and then produce this, and then you generate this kind of feedback loops. Because once you have mechanical heterogeneity, then you initiate systems like flows and contractions, which then again drive morphogenesis, but also instruct the compartmentalization of cell fates.
Yeah, tissue mechanics is so complex. I think we have another question, right? From Srinivas asking, do the fibroblast form an actomyosin ring, like in case of cell extrusion? Oh, like a pinching one, I guess?
Yeah, so like a actomyosin ring in terms of like a supracellular contractile ring. I mean, the fibroblasts they clearly form kind of a ring structure. I don't know, in case of cell extrusion, I'm not quite sure what is the reference, sorry. But yeah, so the fibroblasts form a ring around the placode and they are high in actomyosin. That's what I can say.
All right, great. We have a question from one of our viewers on YouTube. Susan Crawford Young is asking, have you heard of the cell state splitter by R. Gordon? I haven't, but
I have not. So I'm curious to Google that.
Yeah, maybe she can leave a comment giving us a little bit more info or context, and I can pass that along.
I hope I didn't reveal my ignorance.
Sounds interesting. Cell state splitter. So we have another question saying, are there any changes in the expression levels or the organization of the keratins during placode invagination? And what about changes in the organization or expression of the desmosomes or hemidesmosomes?
Right. So we don't see massive changes, but we still need to look more carefully at the keratin organization, because we expect to see some differences, especially if we see differences in compression and this kind of thing. So we would expect maybe some more subtle differences in keratins. we have not looked in the delamination. Obviously, that's very important, and we think, and of course, Kathy Green has beautifully shown that desmogleins, so desmosomal cadherins are definitely important for the actual delamination.
Amazing. There's another question from Sasha Bailey. She's asking, how do cells generally sense this density? Is this via lateral compression, or how did you measure it experimentally? So it's more of a clarification.
Yeah, so that's of course something that we are very interested in understanding. I mean, we think that the cells are sensing deformation, and of course there's beautiful work from Matthew Peel and others really showing that cells are potentially using the nucleus as a ruler, and also we have shown that nuclear deformation is sufficient to drive the chromatin changes. So we think that the cells might use the nucleus, but of course also in general cells are very sensitive to changes in volume. So if you have subtle crowding, one might also expect that you have changes in volume, which have been shown to, for example, directly drive changes in translation, for example, to TOR activity. So there could be multiple mechanisms that act maybe in parallel or depending on the nuclear stiffness or depending on the cell type. But we are very interested in understanding really in vivo in the epidermis what could be the sensors of density and how they could then directly link to the cell fate. So that's something that we are really actively pursuing right now.
Especially, I remember the initial ones where you showed that there is an increase in the actin-myosin contractility with the confinement. That pretty much, yeah, my mind went to the nucleus.
Yeah, exactly. So that's what we are testing right now. That is it the nucleus, or
right.
But it could also be changes in cytoplasmic volume.
Mm-hmm.
Great. We have a question from Ruben saying thanks for the talk, and he's curious about membrane fluidity, specifically when the mitotic cells that appear to help mechanical movement. Do you know if or how the membrane fluidity may interfere in the follicle formation?
Yeah, so when I was talking about fluidity, I was kind of referring to a tissue scale phenomenon. So basically, it has been also shown that, for example, if in a jammed epithelial monolayer you have cell divisions, they are sufficiently to locally fluidify. So I was referring to that. Whether now membrane fluidity, that's kind of a little bit different scale, and we have not addressed that. That's also very, very difficult to address in vivo. But of course, that would be super interesting to see whether membrane fluidity, membrane tension, I mean, we already discussed that a little bit earlier in terms of signaling or mechanics could affect. But yeah, so far we lack the tools to really address this in our system.
All right, fantastic. We have another question, again, from YouTube. Wondering about when you talked about your 3D vertex model, specifically what tensions were changed in order to allow certain cells to extrude?
Right. So basically we modulate the interfacial tensions as a proxy for basically the myosin distribution. So we have obviously the lateral interfaces, then we have the basal interface basal interface and the apical interface, and we can independently kind of alter those. And then basically, so in the phase diagrams, I showed that, for example, we modulated the ratio between the apical and basal because we saw the differences in the myosin distribution. And then in parallel with modulating the apical to basal, we modulate, for example, the lateral. And this was sufficient to produce some sort of invagination, but not these oscillatory flows or the full downward invagination. And for that, we then incorporated, in addition to changing the interfacial tensor, we kind of inserted velocities, basically, that came from the adjacent cells towards the placode as a proxy for extrinsic forces. So that was then not modulating interfacial tension, but basically inserting additional directional velocities. Yeah, and sorry, Lisa probably would explain the model much better than I would, but I hope this was a good enough answer.
No, that's fantastic. Thank you so much. All right. Okay. We have more questions coming. I keep getting ready to wrap it up. But that's great. I'm glad people are so excited and curious. Anka, did you want to take the next one?
Yeah, sorry. Yeah. Another question from Luke asking, once the placoid formation has taken place, and is it enough to establish classical polarity signal like PCP in the placoid to give directionality? A lot of symmetry breaking and PCP.
Yeah, so indeed, I didn't go into this, but already at E15, when we see the downward flows, there's also polarity, which is planar cell polarity, because the fact that we can comb our hair is because all the hair follicles are oriented through planar cell polarity, and this already happens at that stage. And we do see, for example, that the fibroblasts also start to be polarized. So there is clearly already establishment of planar cell polarity, and possibly also the flows start to be directional. But we have basically not tried to address this aspect in our current work or in our model, but that's of course fascinating. And for example, Danielle Davenport has done really fantastic work on that. And there's also an interesting interplay between mechanics and basal cell polarity. So the planar cell polarity, so she has, for example, shown that basically global tissue strains are participating in orienting this. So there's also kind of feedbacks between the mechanics and the PCP. But that's already pretty complex. So it's maybe the next step in our work as well, to try to address how then these polar flows are starting to form.
All right, awesome. Thank you so much. I think that may actually be the end of our questions. I will, okay. Yes, and it looks like your question from about the vertex model was well answered. They said, thank you so much. All right. With that, I think we will wrap it up. So again, thank you to Sara for her fantastic end of the season seminar. To those of you watching online later,